Our new chapter is going to focus on genetics. So we are going to talk about how you inherit different types of traits. So this will include talking about Mendel, Gregor Mendel, the father of genetics, and what he discovered, how chromosomes are involved in your genetic traits, how some traits are linked, what are called sex-linked traits, and are carried on what are called the sex chromosomes. And we'll also talk about some exceptions to some of the things Mendel discovered, some traits that he wasn't aware of during his time. So let's start with an introduction. So your heredity is the transmission of traits from one generation to the next. So what you inherit. For example, whether you have brown hair, or whether you have curly hair, or blood type A, or if you're lactose intolerant. And genetics is the study of heredity. Now, any feature that you can inherit that varies among individuals is called a character. For example, hair color would be a character. Blood type would be a character. In pea plants, because that's what Mendel studied, flower color was a character. And then the traits are the individual variations. So for example, for the character of hair color, you might have brown or black or blonde or red hair. For the character of blood type, you might be blood type A or blood type B or AB, etc. And in the pea plants, flower color could either be purple or white. So those were different traits, and the character was the general characteristic that had these variations. So Gregor Mendel, he is considered the father of genetics because he was the first one that quantitatively tracked traits, meaning numerically, over several generations. And he is the one that figured out, although he did not use this vocabulary, he's the one that sort of figured out that your genes carried these versions of traits, which we today call alleles. And he came up with 3 principles. Now, one thing you need to understand when we get into Mendel is you need to understand how he named the generations. So the original purebreds that he would cross would be called the P generation. You can actually think for P, it stands for parental, the original parents. And he always started with purebred parents. So in this case, you see a picture with pigeons. He was studying pea plants. But these would be in the P generation, these would be the purebred pigeons. So maybe a white one crossed with a gray one. Their offspring are called F1. Maybe like YF. It stands for, the 1 is for first, and the F is for filial. Filial means offspring or sons. And then what he did was he would cross 2 of those with each other, which seems kind of gross with pigeons, but with flowers, not such a big deal. Cross 2 of these with one another, and they would give you F2, which the 2, it means it's the second filial generation. And you can kind of think of this as sort of like, grandparents, parents, and then the children. So this is how he always did his traits. He always started with purebred parents, crossed those and got an F1, crossed the F1 and got an F2. So he was actually a monk, and he worked in the garden, and so he worked with pea plants because they were available to him. And what he did, so if you look at the flower from a pea plant, you'll see that it's got both female and male parts. Oh, sorry. So the carpel is the female parts, that's where the eggs are, and the stamen are the male parts. So you've got male parts here, and then you've got a female part in the middle. So theoretically, they could fertilize themselves, which he would not have wanted to do, because if they fertilize themselves, you basically have purple flowers fertilizing something that also is carrying the trait for purple flowers. So the way that he crossed these, he cross-pollinated. So if you look at the picture here, he basically took the male parts off of this purple one, and then took pollen, which is basically the sperm in the pea plant, from a white flowered plant, and fertilized the purple flowered plant. And that way he could get a hybrid, or his F1 would be a mix of the purple and the white. Now, people in his time period thought that traits blended. They believed in blending inheritance, which just basically meant if you crossed purple and white and the offspring came out purple, white was just gone forever. That's what people believed. And he actually showed that that wasn't true. So he crossed these, they developed into peas, he grows them into a new set of plants, and then he could observe the offspring, the F1, and in this case you can actually see that they all had purple flowers. These are all of the traits that he tested. So you could see purple versus white flowers, axial versus terminal, meaning the flowers are in the middle of the plant versus at the tip. The peas could actually be yellow or green. They could also be either round or wrinkled. The pea pods can either be inflated or they could look kind of wrinkly and constricted. They could be green or yellow, and the plant could either be tall or short. Every single trait that he tested had exactly two versions. Purple versus white, tall versus short. There was nothing in between. There wasn't anything weird. And if there had been, he probably would have been really confused. But because pea plants were so easy to study, they are quick to breed, make lots of offspring, and the traits are easy to observe, he was able to come up with his principles. So they actually show 4 different hypotheses here. The first one is that there's different versions of genes called alleles. So in this case, to apply this new vocabulary, there would be one gene for flower color, but it had 2 alleles, 2 versions. There was one that coded for making purple flowers and one that coded for making white flowers. So that was the first thing. The second thing he said was that every organism gets 2 copies of a gene, one from each parent. So in his head, he's thinking, okay, so this one, that's purple, and this one is white, and each of those has 2 copies of the information. So let's just call them big P, and little p. And he's saying that each parent is going to donate just 1 copy to the offspring, and the offspring will therefore have 2 copies in the end. So they got 1 copy from mom, 1 copy from dad. The third thing that you should know that's related to this is that if the 2 copies are identical, we call that being homozygous. Homo means the same. But if the 2 copies, the 2 alleles are different, we call it heterozygous. So if, and I was using letters, and we're going to continue to use letters for this, so if the organism was big P, or little p, these were the purple flowered plants, and this is what we were using for the white flowered plants, we called that homozygous. If they had 2 different versions, so they were big P, little p, we call that heterozygous. So to be heterozygous, it has to be a capital and a lowercase in this scenario. Now, why am I using capital and lowercase? We're going to get into that. So it turns out that if you did have an organism that was heterozygous, 2 copies, the appearance would be determined by the dominant allele. In other words, in every trait he tested, one of the traits covered up the other. So, for example, with the flowers and the peas, it turned out that purple was dominant to white. So big P, little p was purple in color. It doesn't mean they weren't carrying the gene for white, but they were purple. And not only that, but that means that the ones that were big P, and the ones that were big P, little p would both look exactly the same. So he's the one that came up with, we actually call this the principle of dominance, the idea that some traits cover up others. And dominant basically means that if you have a copy of that one, that's the one that's going to show. You're not actually going to see the recessive trait. And so this is explaining his other law, which is called the law of segregation. So we just talked about meiosis, and he didn't know about meiosis, but in essence, that's what's happening in meiosis. If we imagine that these are carried on chromosomes, and you had a parent that was big P, little p, when they go to make sperm or eggs, meiosis would happen, right? The X's would split, and this one would get a big P, and this one would get a little p, and then technically this would split again, if you remember meiosis. So both of these sperm would have big P, and both of these sperm would be carrying little p. And that's the principle of segregation. Segregate just means to separate. And he basically didn't know about meiosis, but he was saying that during meiosis, the genes separate from one another. So here is sort of an overview of what he did. So he crossed purple and white, and they all came out purple. So that tells us purple must be dominant. If you cross 2 of them, whichever one comes out, that's the dominant trait. He assumed these were purebred, so they would have been homozygous, big P, little p, and that means all the offspring would have been big P, little p, because they would have all gotten a big P from one parent and a little p from the other. Now the weird thing is when he crossed two of these, so big P little p with another big P little p, this is where he showed blending is not what happened. Some white ones came back. About a fourth, 25%, came out white. So, to summarize, whatever he saw in the F1, he knew that was the dominant trait. It sort of covered up the other version of the gene. But when he crossed the F1s with one another, he got about 75% purple, 25% white. And we see this with pretty much all of the traits that he studied. Every single time, one of them was dominant to the other, and then if he crossed two of these heterozygous ones together, he always got that same basic ratio. About 3/4 of the offspring had the dominant trait, and about 1/4 showed the recessive trait. All right, so some checkpoint vocabulary. So a gene is a code for a trait. Recessive allele, we're going to use a lowercase letter. We're usually going to pick one letter for the gene, and we're going to use the lowercase letter for the recessive allele. We're going to use a capital letter for the dominant allele. I'm just using big B and little b. In the case of the flowers, we used big P and little p. We used P for purple because that was the dominant trait. Homozygous or true breeding or purebred means the two alleles are the same. Big B or little b. Homozygous dominant specifically means big B. Homozygous recessive, little b. If they just say homozygous, it could be either one. And then heterozygous is always big B, little b. The genotype, these are new words. The genotype is the specific gene combination. And the phenotype, I always tell people, think genotype genes. What do the genes look like? Phenotype, what do they look like physically? So think phenotype, physical appearance. And so the physical result being black, white, curly hair, straight hair, blood type A, that's the phenotype. Okay, so every diploid cell has the chromosomes in pairs. We've already talked about this. This is sort of a refresher. The chromosomes carry alleles for the same genes at the same locations. And we talked about this too, that, for example, if chromosome 9 carries the gene for blood type, and this is me, I would have 2 copies of chromosome 9, and maybe one of them codes for blood type A, and maybe the other one codes for blood type O. But in that spot on chromosome 9, that's where I would have that gene for blood type. So every animal or organism, will have 2 copies of every gene. They could be the same or they could be different from one another. And then during meiosis, that's the process where randomly one copy only is going to be given to a sperm or an egg. And that's the law of segregation. So we just kind of went over this, but this is just applying it to our previous chapter. All right, and that brings us to how Mendel explained the 3-to-1 ratio. How come in the F2, the children of the children, how come they came out 3/4 showing the dominant trait and 3/4 with the recessive trait? And so that comes from this principle of segregation. So let me show you. So the original parents were big P, and little p, right? That means that they only had one option during meiosis. So this one goes through meiosis and randomly gives something. The only option is capital P. The other parent goes through meiosis. The only option is little p. And that means the offspring, there's only one option, big P, little p, which we just said would be purple. But the F1s were hybrid or heterozygous. They were big P, little p. So what happens when it's heterozygous? Well, that means that during meiosis, parent number 1 can either give big P or little p. And parent number 2 could also give big P or little p. So what are we going to get in the offspring? Well, we could get big P, right? Or we could get little p. Or we could get big P from this parent and little p from the other parent. Or we could get big P from this parent, and you always write the capital letter first, by the way, and little p from this parent. So you get basically 25% chance of getting this, a 25% chance of getting this, and actually a 50% chance of getting this. And what would these look like? Well, purple, purple, purple. Hey, look at that. 3/4 would be purple. Now there's a way better way to do this, and that's with a Punnett square, which is what we're going to talk about tomorrow, using a Punnett square.